Good morning, and welcome to CROI 2020. I'm Jim Hoxie, and along with Wafa El-Sadr, and on behalf of the Scientific Program Committee, welcome you to the first plenary session of this meeting. Before we get started, I have a couple of announcements. On Tuesday at noon, there will be a special session from 12:10 to 1:20 Eastern Daylight Time on the epidemiology and virology of the coronavirus outbreak, featuring Drs. Zunyun Wu from China CDC, Ralph Barrick from UNC, John Brooks from the US CDC, and Tony Fauci from NIH. On Wednesday at noon, and this is a change from the first program that was distributed, there will be a special session providing an update on the HVTN 702 vaccine trial, featuring Drs. Larry Corey from the Fred Hutchinson Cancer Research Center in Seattle and Glenda Gray from Johannesburg, South Africa. In today's first plenary talk, you will hear about the progress and challenges towards the development of a cure for HIV infection. Since the remarkable cases of Timothy Ray Brown, first reported 10 years ago at CROI, and of the London patient presented last year at this meeting, we know that curing HIV is feasible. However, a safe and scalable cure for 38 million people living with this virus has not been realized. There are many scientific and ethical challenges that we face. HIV persists in viral reservoirs that can last for the lifetime of people living with HIV. Although the cells that harbor HIV are rare, they represent a long-lasting source of virus and are the reason why antiretroviral therapy must be taken for life. Our speaker today is a leader in the characterization of HIV reservoirs and in the development of therapeutic strategies for their elimination. Dr. Sharon Lewin is the director of the Peter Doherty Institute for Infection and Immunity in Melbourne, Australia. Sharon is an infectious diseases physician and basic scientist who has made numerous and highly significant contributions to the field of HIV cure. I am sure you will appreciate why she is so respected in this area of research. As a basic scientist, Sharon made important discoveries in HIV latency and the molecular mechanisms that are involved in HIV persistence during antiretroviral therapy. But since she is also a clinician, Dr. Lewin always thinks translationally and is well recognized for her ability to bring basic discoveries from bench to bedside, as you can see by the title of her presentation today. Connecting is a key word for Dr. Lewin's research style. She is a leader in establishing collaborations with groups in the US, Europe, and Asia. And her work and collaborations have spanned and linked basic, clinical, and social sciences. Dr. Lewin is the perfect example of a scientist involved in HIV research at many different levels, not only from the basic science and clinical aspects of HIV cure research, but also by being an advocate for people living with HIV. Dr. Lewin is an inspiration to many, and I'm very pleased to have the honor of introducing her as the first plenary speaker of CROI 2020. Please welcome Dr. Sharon Lewin. She has nothing to disclose.
Thank you very much for the kind introduction, Jim. It's a great honour to be delivering this talk, even if it's virtual, and I'm sitting in my office in the middle of the night in Melbourne, but at least it's the middle of summer. I wanted to congratulate, first of all, the CROI leadership for amazing work in extraordinary circumstances over the last few days. And the HIV community has been through extraordinary times before, and we'll get through this, and I'm sure CROI 2020 will be a very memorable meeting. My talk will be in three parts. I'm going to first review advances in our understanding of where and how HIV persists on antiretroviral therapy from laboratory studies. I'll then discuss clinical strategies being evaluated now and in the future and some associated challenges. But finally, I'd like to emphasise that our work in HIV cure research can't be done in isolation. We need to continually work with people living with HIV to understand their preferred characteristics of an HIV cure product and ensure that we're thinking now about how a product will ultimately be delivered in the field. I thought I'd start with a brief history of HIV cure research and some important examples of cure and remission and where we are now in 2020. We first realised that HIV couldn't be cured in 1997 with the discovery of HIV latency. 12 years later, there was tremendous excitement with the description of the Berlin patient, or Timothy Brown, who was cured from HIV following a bone marrow transplantation. The following year, we became aware that some individuals are able to control HIV after a period of being on antiretroviral therapy, what's now called post-treatment control or remission. And in 2013 were the first case reports of individuals with extremely small reservoirs from very early antiretroviral therapy or other forms of transplantation who had no detectable virus but then had viral rebound months to years after stopping antiretroviral therapy with an unexpected time course. And then last year, as you heard earlier, the second report of a man cured of HIV, the London patient, as well as the Dusseldorf patient, and we're going to hear updates on both these exciting case reports at CROI this year. So where are we in 2020? Well, latency is far more complex than we initially thought in 1997. Combination approaches that target the virus and immune system will almost certainly be needed. And it's important to keep in mind that the why and who needs a cure for HIV is changing. So why and how does HIV persist on antiretroviral therapy? Well, HIV largely established itself in two forms. First of all, there's productive infection, where virus infects an activated CD4 T cell, can be detected as DNA positive, RNA positive, HIV proteins are found on the surface of the cell, and these cells usually die. In contrast, latent infection is characterised by integration of the viral genome into the host genome and is detected as DNA positive. However, this genome is transcriptionally silent and RNA negative. No protein is expressed on the surface of the cell. And these cells, we now know, are primed to survive. But HIV latency is not binary. There's actually a spectrum of activity ranging from productive to latent infection. So I'm going to first review 4 new concepts in HIV persistence and latency. First, the concept of reservoir activity. Second, the importance of proliferation. Third, the importance of position, where HIV integrates into the genome. And finally, why it's important that we can quantify both defective and intact virus. So reservoir activity is dependent on a number of factors. First of all, the importance of place. We now know that tissues such as the lymph node, gastrointestinal and female genital tracts not only have more infected cells, but these cells are more likely to be RNA positive or transcriptionally active. In addition, these tissue sites are enriched for specific T cells that are permissive to infection and persistence, so-called tissue-resident memory cells. Reservoir activity is also dependent on time. Several years ago, my group demonstrated that proteins that control the sleep-wake cycle of circadian rhythm, CLOP and BMAL1, can also directly activate HIV transcription. And in a prospective study of people living with HIV on antiretroviral therapy, we demonstrated that HIV RNA expression actually varies with time of day, consistent with the circadian cycle. And in this study, you can see here the measurement of a classical circadian protein, salivary cortisol, showing variation of the 24-hour period. And we saw similar variations of HIV RNA and the ratio of RNA to DNA, but no change in the number of infected cells. And this work is important because it has implications for when and the time that we measure the viral reservoir in clinical trials, and it also gives us ideas about new ways that we may be able to perturb latency. Proliferation has also become a very important concept, leading to HIV persistence. We used to once think of HIV just infecting long-lived cells, such as naive or memory T cells. But we now know that infected cells can proliferate, and these daughter cells contain the same virus in the identical integration site. It's also now well described that clonally expanded infected cells make up about 50% of the reservoir. These clonally expanded cells can contain intact virus and contribute to plasma virus rebound after stopping antiretroviral therapy. But what we don't currently fully understand is the drivers for proliferation. Potential explanations include homeostatic proliferation, a basic feature of T cell biology, antigen-specific expansion, or whether this is driven by HIV itself through the sites of integration. This work I'm going to show you was done in collaboration with Peter Hunt and Charlene Bacchus-Soufan from UCSF, where they enrolled people living with HIV on treatment for at least 2 years, labelled their cells with deuterated water, and then sorted cells into T cell subsets. And we measured the percentage of these subsets that contained infected clones. And what I'm showing here is that the percentage of cells that had integration sites that were clonally expanded was significantly enriched in effector memory T cells compared to less differentiated T cells. And as effector memory T cells are T cells that respond to antigen, this supports the concept that proliferation is driven by specific antigens. And at the meeting this year, you'll see several presentations on this concept, and I draw your attention to 2 presentations from Simonetti et al. And Mendoza et al., looking at the direct effect of antigens on T cell proliferation. Position matters. HIV integration occurs in distinct sites in both clonally expanded cells and cells that have single integration sites. This work was done on the same cohort from UCSF by Yori Simons, a postdoc in my group, together with Simon Malone from Perth and now Vanderbilt University, where they used a high throughput assay to compare integration sites in clonally expanded cells compared to singly infected cells. And this diagram shows you a number of genes, each gene in a separate row, distinguished by whether they were found in single or clonally expanded cells. And the intensity of colour shows you the frequency of that integration site. And what you can see quite clearly is the marked difference in integration sites in clonally expanded cells compared to those with single integration sites. And finally, when we looked at the percentage of genes that had integration that had integration sites associated with cancer, these were also markedly expanded in clonally expanded cells. Position matters also in how active the virus is. Integration sites determine the likelihood of a virus being active or silent. And it's interesting that intact viruses are even more likely than defective viruses to be integrated in sites which keep the virus silent, another mechanism for persistence of intact virus. And finally, quantification of defective and intact virus is extremely important in understanding the reservoir. And we now have new assays that can rapidly detect intact virus, essentially the virus that matters or capable of rebound. These assays are done by PCR with primers that detect intact and defective DNA and now allow for large natural history studies and for the use of these assays in clinical trials. This is one example of a natural history study from Steve Deeks that looked at the decay rates of intact compared to defective virus over time in individuals on antiretroviral therapy. And what you can see here is that there is clear decay of the intact virus not seen with defective virus, raising hopes that intact virus may indeed decay over time. And in fact, elimination of intact virus may be what you might consider a new definition of cure. Several years ago, a subgroup of elite controllers were given the term exceptional elite control. These were people that have never received antiretroviral therapy, but had very infrequent detectable DNA. Recently, Yu Xu has extensively studied an exceptional elite controller, Lorene Willenberg, and a woman living with HIV in San Francisco, never been on antiretroviral therapy, and who's had undetectable plasma HIV RNA for over 24 years. And you looked at a very large number of cells from both blood and the gastrointestinal tract and used a range of approaches, including full-length sequencing, viral outgrowth, and detection of intact proviral DNA. And although it could find remnants of viral sequences, could find not one intact virus in all of these cells studied. So in fact, when we think about cure, we may not eliminate every viral genome, but what we aim to do is eliminate intact virus. So what are some of the clinical strategies being tested currently, and what are the challenges associated with these strategies? Well, the overarching goal of cure strategies is to essentially eliminate this pool of latently infected cells. If we could eliminate every cell, this would essentially be what we'd call eradication. But if we could just reduce the number of infected cells and maintain control through immune-mediated mechanisms, this would lead to what's now commonly called remission. Now, nearly all strategies involved at eliminating the reservoir either target the virus or immune system. There's a range of strategies specifically aimed at eliminating viral infected cells and a range of strategies that boost immune function. Nearly all of these have now been tested in human clinical trials, except these highlighted here in green. And over the next few minutes, I'm going to focus on advances in some particular areas, specifically latency reversal, the use of broadly neutralising antibodies, immunomodulation, CAR T cells, and gene editing. So we've known about a large number of latency reversing agents for many years, and several of these agents have now been in clinical trials. And the conclusion from these clinical trials are that these drugs are able to shock, perhaps not strong enough, but not yet kill. So there's a need for more potent and less toxic latency reversing agents. There's a need for more approaches to get the kill into shock and kill. And one of these approaches could be the use of pro-apoptotic drugs. And finally, some of these latency reversing agents also have immune modulating activities, which could be potentially quite beneficial. Example of these including TLR agonists, which you'll hear more about at this meeting, or anti-PD-1. And I'm going to talk in a little bit more detail about a new class of latency reversing agents, SMAC mimetics, some recent data on immune checkpoint blockers, and a reference to TLR7 agonists. So a couple of months ago, there was some exciting work published on a new class of latency reversing agents that were more potent and less toxic than any other agents yet investigated. These drugs are from a class called SMAC mimetics. They were developed for cancer to inhibit proteins that block apoptosis. However, they also activate NF-kappa B, a key transcription factor that drives HIV production. And in animal models, both mouse and macaque, a particular SMAC mimetic, audD2a, showed potent activation in blood, shown here as recurrent blips in SIV-infected macaques on antiretroviral therapy, as well as an increase in RNA in lymph node tissue as evidenced by an increase in SIV RNA. And these potent effects were not associated with any adverse events or toxicity. And these drugs are yet to be studied in human clinical trials. Another potential attractive latency reversal agent are agents that could potentially do both together, both activate the immune system and reverse latency. And immune checkpoint blockers are agents that could potentially do both. We've known for many years that latent virus is enriched in cells that express immune checkpoint markers, particularly PD-1 and other markers including CTLA4 and TIGIT. In vitro, ex vivo and in vivo, anti-PD-1 can reverse HIV latency, and we've recently shown that the effect is even greater when used in combination with anti-CTLA4 in vitro. Anti-PD-1 can also increase antigen-specific T cell function, leading to enhanced viral control in macaques. But probably the biggest concern with these agents is how we're going to get them into the clinic due to the significant challenges related to immune-related toxicity. However, this could potentially be overcome by optimisation of dosing and use of new agents that are now appearing that don't use antibody function but potentially other strategies. And I draw your attention to 3 presentations at CROI, one from Thomas Rasmussen and one from Jill Lau from my group, who are both investigating combination immune checkpoint blockade in vivo, and a presentation from Afa Makoyi, who's looked at anti-PD-1 and its effect in causing post-treatment control in a macaque model. Finally, it's unlikely that any of these agents will work alone. We need to target the virus as well as the immune system together at what's commonly called combination immunotherapy. There are many promising studies from macaque models showing that combination immunotherapy can indeed lead to cure or prolonged remission. Combination immunotherapy in human clinical trials are really only just starting as large and or randomised clinical trials. The first trial to be reported was RIVER, a trial that included a strategy to reduce the reservoir or latency reversing agent vorinostat together with an HIV-directed vaccine with a chimp adenovirus. In RIVER, they saw no change in the reservoir. However, the clinical trial did not include a treatment interruption, so it's unclear what would have happened if antiretroviral therapy indeed was interrupted. There are now many studies combining interventions to both reduce the viral reservoir and increase immune control. And one of these important new studies will be presented at CROI later today, looking at the combination of romidepsin with the broadly neutralising antibody 3BNC. And there are many other trials that are combining broadly neutralising antibodies with other immunomodulatory agents such as interferon alpha, IL-15, TLR7 agonists and other approaches. And later this morning, you'll hear from Devi Sengupta, who's evaluated the effect of TLR7 agonists in combination with an antiretroviral treatment interruption with some very interesting findings. Finally, gene therapy holds quite a significant promise with the prospect of true eradication. There are a number of ways that gene therapy could potentially eliminate or allow for cure. First of all, gene therapy can lead to what's called attack or enhanced anti-HIV immune responses. Gene therapy can also protect or engineer uninfected cells to be resistant to HIV. And finally, gene therapy can be used to purge or directly eliminate the virus itself. One of the major challenges, though, for gene therapy is its delivery. Currently, delivery occurs largely ex vivo, which means gene editing of cells outside the body. But there's huge hope that one day we'll see more interventions that can deliver gene editing in the body or in vivo. And this is already happening for gene therapy strategies that target the eye and the liver. Many of you will, one approach to gene therapy is the modification of CD4 T cells or NK cells to form CAR T cells. CAR is a chimeric antigen receptor that will bind and kill cells that express HIV envelope, potentially a variant of a broadly neutralising antibody or a CD4 molecule, is forming the ligand binding domain. Binding of this domain to its target then triggers a signal leading to activation of the T cell or NK cell. There are CAR T cells for HIV being tested in mouse and macaque models and already in clinical trials in China and in the US. Some of the major challenges include toxicity, which is now thought to be potentially preventable, delivery to tissue sites, and the low expression of HIV envelope on antiretroviral therapy. Many of you will be familiar with the concept of ex vivo gene therapy that was performed several years ago to modify CCR5 positive cells. However, there are many other multiple phase 1-2 clinical trials now underway that target populations of people living with HIV, both with and without cancer, that target genes such as CCR5, but also HIV, and are also directly targeting different cellular sources, CD4 positive T cells and CD34 positive stem cells, as recently described in a case report from China in the New England Journal of Medicine. However, a very real major advance will be needed in order to get gene therapy in vivo, but this is now happening as well. In vivo gene therapy relies on the delivery of genes directly into the body through either a vector such as adeno-associated virus or a nanoparticle. And already this is happening, delivering genes that code for broadly neutralising antibodies. A report late last year using AAV1 delivering genes to synthesise an antibody PG9. And I draw your attention to a late breaker abstract here at CROI looking at the delivery of BRCA07 antibody genes delivered by AAV8 with potentially successful outcomes. Finally, what are the endpoints for clinical trials currently and what are their challenges? Well, at the moment, the only way we can tell if someone is cured is by treatment interruption, looking to either a delay in viral rebound or post-treatment control. There are obviously significant challenges with allowing individuals to become viremic, both for the participant and for the risk of transmission of virus in this period. I draw your attention to some consensus guidelines on recommendations of how best to run these studies, which are very useful for clinical investigators and relevant ethics committees. Ideally, it would be fantastic if we had a biomarker that you could measure on antiretroviral therapy that could predict the outcomes following treatment interruption, but we currently don't have such a biomarker. An alternative, additional, helpful tool could be if we could measure changes in vivo that occur prior to detection of viremia, but after stopping antiretroviral therapy. And indeed, I was recently involved with a very nice study led by Liddy Troutman looking at this very issue in a cohort from Thailand undergoing treatment interruption studies, and identified one biomarker of activated dendritic cells that were clearly detectable several days before viremia was detected. Finally, in the last few minutes, I want to talk about how we might one day consider implementation of an HIV cure. So this is where we are currently in 2020 at excellent daily treatment modalities. In the next few years, we're almost certainly going to see advances on antiretroviral therapy, long-acting ART, or broadly neutralising antibodies that people might be able to take just 2 or 3 times a year. So what cure modalities might we first see in the clinic? Well, I'm looking towards the potential of us first seeing combination immunotherapy, not leading to cure, but potentially HIV remission. This could then be followed by ex vivo strategies such as gene or cellular therapy. And one day, the hope that we could deliver an in vivo cure potentially through gene therapy. And the overarching goal of all this work is potentially one day a single shot cure that would be available to everyone living with HIV. It's important that we now think about what that target product profile might look like, particularly for delivering a cure in a low-income country. Target product profile is a description of what a product might look like that will be useful, but also that will be possible. And together with the Bill and Melinda Gates Foundation and the International AIDS Society, I've been involved with a large project gaining input into what a TPP for a cure might look like. And this involves engaging all stakeholders to define a cure product that people living with HIV want delivered to the field. It will involve defining the minimally acceptable and optimistic characteristics of a cure in relation to a whole number of parameters. And this target product profile should be dynamic and will evolve based on breakthrough scientific developments and patient needs, but the conversation needs to start now. So in summary, I've explained to you that there are many new concepts in understanding HIV latency, and understanding the basic science that drives HIV persistence will one day lead to better and novel approaches. Combination immunotherapy interventions look promising in monkey models, and we're currently underway with clinical trials to see if similar results will be seen. More invasive and complex interventions should be explored, such as CAR T cells and gene therapy, as there's the potential for eradication. And finally, approaches to an HIV cure will need to adapt to the treatment landscape, the needs of the individual, and to always ensure we have maximum public health impact. Early engagement of community, regulators and all funders will be needed. I'd like to acknowledge a very large team that I lead in Melbourne, Australia. Some fantastic people, much of their work I've shown today. I'd also like to acknowledge the DARE family of investigators, a group I've been part of for the last 8 years, fantastic people doing fantastic science. I'd like to acknowledge a whole range of other collaborators, particularly my clinical colleagues at the Alfred Hospital, and also the people living with HIV, especially in Australia, who have participated in many of the clinical trials we've done. Finally, I'd like to acknowledge the many funders that have supported the work I've presented. And I'd like to acknowledge colleagues, friends, and my lovely family, my 2 boys, Alex and Max, and husband Bob, and my mother Eva, and all of my colleagues who kindly provided comments on the slides I presented today. Thanks very much and enjoy CROI 2020.
Good morning. I'm Wafa El-Sadr from ICAP at Columbia University, and it gives me great pleasure to introduce our second plenary speaker this morning, Dr. Kevin De Kock. Dr. De Kock is a distinguished public health expert and global health leader. And over his career, he's held critically important positions. Most recently, he led the Ebola response for the U.S. Centers for Disease Control in the Democratic Republic of Congo. And until the mid-2019, he was the director of the CDC in Kenya. Prior to that, he was the founding director of the Center for Global Health at the CDC. And he also held the position of director of the World Health Organization Department of AIDS HIV. He has also served as the director of the CDC Division of HIV/AIDS Prevention, Surveillance, and Epidemiology. Over his career, Dr. De Kock has garnered enormous breadth and depth of training and expertise. He obtained his medical degree from the University of Bristol in the UK and specialized in internal medicine as well as in infectious disease, tropical medicine, and liver disease. He is a fellow of the Royal College of Physicians in the UK, and he's also as well has held professorships both at the London School as well as the University of Nairobi. Dr. De Kock has received numerous awards over his career, including the Chalmers Medal from the Royal Society of Tropical Medicine and Hygiene, as well as the Commandeur de l'Ordre de la Santé Publique in Côte d'Ivoire. And most recently, he also received the CDC's Medal of Excellence and Distinguished Service Award for his work in responding to the West Africa Ebola crisis. For all of us who are engaged in the HIV response, whether we're basic scientists, clinicians, or clinical or public health researchers, we always recognize Dr. De Kock as someone who brings very thoughtful insights that challenge as well as advance our thinking. Therefore, we're very privileged this morning to have him present to us on universal test and treat, lessons from the past and for the future. Thank you. Welcome, Dr. De Kock.
Kevin De Kock here, recording the CROI 2020 plenary. Colleagues, friends, good morning. It's a privilege to speak to you. My comments are my own and only my own. The Danish philosopher Kierkegaard observed that history is written backwards, but life must be lived forwards. I will discuss evolution of 3 parallel themes: understanding of HIV viral load, treatment as prevention, and treatment guidelines. Mathematical modeling influenced thinking and the concept of ending AIDS. I'll review the major trials of test and treat, assess progress towards global HIV control, and comment on general implications. Let me start with acknowledgements. Investigators Diane Havler, Richard Hayes, Shaheen Lockman, François Dabis, and Jan Moore generously shared insights. And I thank diverse colleagues, Peter Young and Emily Zelensky in Kenya, Andrew Phillips, Brian Williams, and others from different organizations, and especially PEPFAR. That combination antiretroviral therapy reverses HIV disease was first highlighted at the International AIDS Conference in Vancouver in 1996. Mellors showed viral load predicted HIV progression, and Coffin presented his stark analogy of HIV as a train crash, with viral load as speed and CD4 distance to the crash. Viral load was rapidly shown also to be the major determinant of all modes of HIV transmission, such as from mother to child in Thailand on the right, and in heterosexual couples in Uganda on the left. With better treatment, prevention practice increased focus on HIV testing and on persons with HIV, so-called prevention for positives. Treatment as prevention was demonstrated in 1994 with ACTG-076, showing reduced mother-to-child transmission with zidovudine monotherapy, and in a neglected paper by Musico the same year, by reduced transmission in discordant couples with the infected person on zidovudine. The landmark HBTN-052 trial showing 96% reduction in sexual transmission with combination ART, and conclusive demonstration that undetectable does equal untransmittable came later in 2011 and 2018, respectively. Discussion and controversy were generated by our modeling paper in The Lancet in late 2008 that suggested that universal HIV testing with immediate ART would eventually eliminate HIV in a hypothetical epidemic of South African severity, and this paper was widely cited. Around 2011, scientific and political commentaries began to predict the end of AIDS. In 2014, UNAIDS launched its fast track and the 90-90-90 targets. 90% of HIV positives diagnosed, 90% of the diagnosed on ART, and 90% viral suppression among the treated. 90-90-90 was an advocacy slogan without peer-reviewed evidence, but it became widely interpreted as a programmatic priority and a promise of HIV control. Separate numeric incidence and mortality targets were proposed, although these were not linked to 90-90-90, and these were included in the 2030 United Nations Sustainable Development Goals. And then treatment guidelines. Post-Vancouver, uncertainty about when to start ART resulted in great diversity in guidance over time and place, resolved only 2 decades later by the START and Temprano trials. Therapy, irrespective of CD4, has been the global recommendation since 2015. The Universal Test and Treat, UTT, trials exemplified implementation science, anchored in the real rather than idealized world of classic trials. Implementation science aims to make research potential a reality rather than an aspiration. It is pragmatic, operational, based in the struggle of people's daily lives. And with epidemiology, it underpins the science of global public health. 4 trials were conducted between 2012 and 2017 in Botswana, South Africa and Zambia, Kenya and Uganda, and South Africa. The investigators have formed the UTT trial consortium to compare and synthesize data and conclusions. The studies varied by context, design, intervention packages, data and secondary outcomes, and adaptations to evolving practice. But they all assessed UTT as the intervention of interest, with HIV incidence as primary outcome. If the technical questions were, does UTT reduce community HIV incidence, and if so, by how much? The political question was, will test and treat end the AIDS epidemic? So expectations were high. These were large cluster randomized trials which together evaluated almost 250,000 people out of more than 1 million community participants. Baseline HIV prevalence ranged from 4 to 30%, ART uptake from 30 to 85%. UTT was compared in intervention communities with standard of care in control communities. Search in East Africa and TASP in South Africa, but not the other 2 trials, conducted universal testing in both intervention and control communities. Search reported on multi-disease screening and follow-up for mortality, tuberculosis, and non-communicable diseases. PopArt differed by having 3 arms, not 2. In 2012, WHO was recommending ART at a CD4 count of 350 cells per cubic millimeter and below. Guidance then changed to 500 cells per cubic millimeter, and again in 2015 to treat all, irrespective of CD4. The changing practice is shown for PopArt's 3 arms in the top figure, universal therapy, shown in purple, coming in for arms B and C during the course of the study. The bottom figure shows, on the left, arm A, the original design of UTT plus an enhanced prevention package, arm B, enhanced prevention but standard of care ART, and arm C, standard of care alone. To the right of the arrow, you see that with revised guidelines, everyone received immediate ART. Arm B became identical to arm A, and the control, arm C, differed only by not receiving other enhanced prevention. And these changes in ART practice applied in some way across all of the trials. The results of TASP in South Africa were published in 2018 in Lancet HIV, and of the others in 2019 in the New England Journal of Medicine. The essential results are shown numerically at the top of this slide and graphically below. The table shows the study or arm in the first column, participants evaluated, and in the third column, statistically significant incidence reduction compared to controls with confidence intervals for the rate ratio or equivalent. Search in Kenya and Uganda and TASP in South Africa showed no significant reduction in HIV incidence. The Botswana study showed a statistically significant 30% reduction. PopArt in South Africa and Zambia had mixed results. Arm A, enhanced prevention plus UTT, showed no significant reduction. Arm B, enhanced prevention with standard of care ART, showed a significant 30% reduction versus the control arm, and combined A and B reduced HIV incidence by 20% compared to control arm C. The authors justified pooling because arms A and B were identical for most of the analysis period because of the altered ART guidance. The figure at the bottom shows effect sizes and confidence intervals, with only Botswana and PopArt arm B significant, with confidence intervals not crossing 0. The combined PopArt analysis is not shown. Additional observations include the persistent high HIV incidence. In the intervention arms, this ranged from 0.59 to 2.1 per 100 person years. Sub-analyses showed differential reductions in HIV incidence by sex, generally greater in men, by age, and by location. Search documented 23% reduced mortality in the intervention group, 21% reduction in tuberculosis or death, and 26% increase in blood pressure control. These trials were conducted in environments of generally declining HIV incidence. Search saw an overall decline of 32% across the whole population over 3 years. Search also demonstrated reduced mother-to-child transmission to less than 1% and increased HIV-free child survival. And please see the oral abstract 4528 on Wednesday. The green histograms on the left show 1990 estimates by individual trial, baseline in light green, end of study, dark green. Greater than 90% of serostatus, the first 90, was achieved across the board. For the second 90, TASP suffered from poor linkage, but the other studies achieved 88 to 97% uptake of ART in persons diagnosed. The third 90, viral suppression in the treated, ranged from 87 to 97% at study end. 3 of the 4 studies, therefore, achieved the UNAIDS targets. Not shown, the diagnostic and treatment gap in men and in youth was narrowed, and viral suppression in men approached that in women. Most important, the blue histograms on the right show population-level viral suppression, the product of the 1990 estimates. Excluding TASP, population viral suppression at the end of the studies ranged from 74 to 88%, exceeding the 73% suppression defined by fast-track 1990 targets. The question is why greater or statistically significant reduction in HIV incidence was not observed. Potential reasons include reduced differences between intervention and control communities through extensive community HIV testing, such as in SEARCH, and universal treatment introduced over the course of the trials, eroding the differences between intervention and control communities. Linkage was low in TASP, delayed in some others. Unreached groups could include persons at greater risk or more likely to transmit. That PopArt's original UTT Arm A did not show reduced incidence, while weaker Arm B did, remains unexplained, and justification for the pooled analysis, giving 20% effect size, is questionable. The studies were conducted in contexts of robust AIDS control programs and generally declining epidemics. Perhaps the impressive population viral level suppression achieved is still not enough. The interventions applied to individuals, but outcomes were measured in populations, and population effects are always lower than individual results in clinical trials. And perhaps mathematical models were too idealistic. The statistician George Box famously observed that all models are wrong, but some are useful. I'm with that epidemiologist, Bob Dylan, when he said that it don't take a weatherman to know which way the wind blows. And there is no doubt that Brian Williams's model behind the Lancet paper was useful. There is a unifying hypothesis pulling this together, and it's that HIV incidence is determined by the prevalence of unsuppressed virus in any community, and that incidence can be reduced by increased testing and treatment. This is supported by the collective data. The tableaus on this slide show strong associations between HIV incidence and population prevalence of unsuppressed viremia, albeit in different axes and scales in the different figures. But you see the straight lines across all of the data. On the left, data from the UTT trials, top right, PopArt, and bottom right, experience among men who have sex with men in New South Wales, Australia. And another shout out, if I may, abstracts 3967, 3173, and 2787 at this conference. So I conclude that these trials were successful. They answered the questions posed, and much clarifying information will be forthcoming, including phylogenetic, social science, and economic data. 1990 targets were rapidly achieved with high population level viral suppression. UTT clearly contributes to HIV incidence reduction, but incidence nonetheless remained high. 1990 does not result in HIV elimination, and UTT alone, at least at this impressive level of performance, will not control generalized HIV epidemics on their own. Questions have been raised about what really was learned and whether the investment, probably $200 to $300 million, was useful. It's a large sum, although it's modest in the context of $9.5 billion of development assistance annually for HIV that's shown on this slide, or the approximately $3 billion that NIH alone spent annually on HIV research over this time, and NIH was not the sole funder of these trials. In the currency of research, publications, theses, interdisciplinary collaboration, capacity building, the contribution from UTT trials is substantial. Feasibility and impact of community-based service delivery were demonstrated, thousands of additional people were diagnosed and treated, and longer-term public health benefit awaits measurement. The studies are influencing the work of policy and funding bodies, as well as the unfinished dialogue on the end of AIDS. I personally don't question this research that was conceptualized in an earlier time and with an earlier knowledge base, but I am troubled by the evolution of therapeutic research. Vancouver 1996 showed the miracle of combination ART. Subsequent guidelines on when to start treatment were heterogeneous and frequently changing, an indication of uncertainty and need for randomized trials. That it took 2 decades post-Vancouver to prove immediate therapy is best seems astonishingly delayed. If we'd known for certain that HIV needs immediate ART, guidelines would have been clear and discussion around UTT very different, because every individual needs to be found and treated, and that is where we are today. But history is written backwards. Life is lived forwards. We have much to reflect on, and I'll comment on HIV testing, key populations, the end of AIDS, and the need for epidemiology. Kim and Young in Kenya showed that adjusting self-reported serostatus in surveys for the presence of ART metabolites or suppressed viral load increased estimates of the first 90 by as much as an additional 10%. Nonetheless, the first 90 is consistently the weakest part of the cascade, and we need research, innovation, and flexibility to close the gap. Available African data consistently show disproportionate incidence in young women and diagnostic and treatment need in youth and men. A research question is who is infecting whom, where, and how. Preliminary phylogenetic results from the UTT trials suggest a proportion of incident infections had their source from outside of study communities. When undiagnosed HIV is rare, more testing will be required, not less, in diverse ways in the community and facilities. PEPFAR places undue emphasis on yield, the prevalence of tests that are positive. For universal treatment, absolute numbers of positives matter more than yield. This +1 paper at the bottom right by Joseph et al. Showed that relaxing testing criteria in health facilities in Western Kenya, including for persons accompanying patients, resulted in more positive diagnoses, including in men. Pop-Art would have missed 80% of their positives with only index testing and partner notification. So, focus on absolute numbers, not yield. The UTT trials, understandably, did not prioritize high-risk groups. In 2018, 54% of global incident HIV likely occurred among key populations, 25% in East and Southern Africa, higher elsewhere. A disproportionately small fraction of funding is targeted towards these groups. As generalized African epidemics decline, HIV may increasingly find refuge in persons in the shadows, including migrants. We must look to where incidence will be, not only where it was. Ending AIDS has been poorly defined with too many metrics to review. Williams arbitrarily defined HIV elimination, that is reducing incidence below a certain threshold of threat, as less than 1 case per 1,000 per year. The ratio of HIV incidence to mortality relates to the modeling logic. If we sharply reduce new HIV infections, then all-cause mortality will eventually eliminate HIV as infected persons die without being replaced. Clearly, we need universal ART, and we should not accept that if we cannot treat our way out of the epidemic, then perhaps we can die our way out of it. Under current conditions, we could eliminate global mortality from AIDS, as well as transmission of HIV from mother to child. Whatever metric is used, and the simplest is probably numeric targets, we are not on track to meet global aspirations. Incidence in the UTT intervention arms ranged from 6 to 21 times greater than Williams's elimination metric of 1 per 1,000 per year. And concerning 1990, current estimates suggest 62% global treatment access and 53% global population level viral suppression. The end of AIDS, therefore, is not in sight, but the UTT trials show how much can be achieved in a short time. And we should not forget that extraordinary progress has been achieved in the real world. Reports such as these from Rakhine, Uganda, and Western Kenya have shown important reductions in HIV incidence and mortality. The top figure showing HIV prevalence trends in red and declining incidence in blue in Rakhine, in association with scale-up of male circumcision in green, lower down, and ART in brown. We have three powerful biomedical interventions for prevention in generalized epidemics: antiretroviral treatment, male circumcision, a once-only intervention whose protection is sustained over time, and PrEP, whose impact in generalized epidemics requires further study. How best to combine and evaluate impact of these and other basic services, including condoms, is an implementation science question for this era that is dominated by talk of universal health coverage. We need more real-world epidemiology and surveillance through longitudinal platforms. The shape of the epidemic is determined by HIV incidence and death, only measurable over time through longitudinal evaluations. Though useful, 1990 are process ratios giving insight into neither epidemic forces nor program quality or impact on disease and death. And if I may, a parting thought. The UTT trials illustrate what we see in the current very challenging global health context, but also with individual patients. And that is that no matter how much data at our disposal, how many epidemiologic analyses or clinical tests, we cannot escape the need for good judgment about their meaning and their use. Judgment cannot be cloned or sequenced. It cannot be procured, but our populations and our patients depend on it. Thank you for your attention.
Thank you very much, Dr. DeCock, for your superb presentation. I'd like to invite all of the viewers today to please send your questions and comments directly to Dr. DeCock to the email address that's shown on the slide. At this point, I'd like to close this plenary session and invite you all to join us for the oral abstract sessions that start at 10 a.m. Eastern Daylight Savings Time. Thank you very much.
Go.
Hello, and welcome to the oral abstract session, Therapeutic Interventions for HIV Treatment and Eradication. We're broadcasting to you live from virtual CROI. I'm Judith Currier, here with Dr. Michael Sagg from the University of Alabama, and we'll be moderating this session. We regret that the moderators who were scheduled to be here, Dr. Maria Buzan and Dr. audD2a Rana, do not get the opportunity to moderate the session today. Okay, so we'll start off with our first presentation. The title is Cabotegravir and Rilpivirine every 2 months is non-inferior to monthly monthly therapy audD2m study that will be presented by Dr. Turner Overton from the University of Alabama. His disclosures are shown on the slide. Dr. Overton.
Hello, this is Turner Overton from the University of Alabama at Birmingham. On behalf of my co-authors and co-investigators of the audD2m study, I'd like to thank the organizers of CROI 2020 for selecting our abstract for an oral presentation. Despite the success of antiretroviral therapy, there remains a need for more convenient, less frequent dosing strategies to help issues related to long-term virologic suppression and ongoing adherence, particularly when related to pill burden, pill fatigue, interactions with drugs or food, or stigma related to HIV or HIV therapy. cabotegravir and rilpivirine are currently under development as long-acting injectable two-drug regimen for the maintenance of virologic suppression in people living with HIV. The Phase 3 ATLAS and FLAIR studies, which were recently published in the New England Journal of Medicine, have demonstrated that cabotegravir plus rilpivirine long-acting dosed intramuscularly every 4 weeks was non-inferior to daily oral 3-drug antiretroviral therapy for the maintenance of virologic suppression. Furthermore, data from the Phase 2 audD2a study demonstrated that Q8-week dosing is non-inferior to Q4-week dosing, with 94% of people on the Q8-week arm maintaining virologic suppression at 96 weeks. These data provide rationale for another study to further evaluate the Q8-week dosing strategy. The ATLAS-2M study is a Phase 3 randomized multi-center parallel group non-inferiority open-label study that builds on the ATLAS study to address this very question. Individuals in the ATLAS study, whether they were already on injectable long-acting Q4-week cabotegravir plus rilpivirine or the standard of care therapy, could roll into ATLAS-2M. In addition, participants could be enrolled if they were on a stable 3-drug regimen with 2 NRTIs plus a third agent if they had no evidence of virologic failure, major integrase inhibitor or NNRTI mutations, and had virologic suppression. Individuals were randomized 1-to-1 to receive either Q8-week dosing or Q4-week dosing of cabotegravir plus rilpivirine long-acting intramuscularly. For those individuals on oral therapy at the time of randomization, a 4-week lead-in period with cabotegravir and rilpivirine were given to assure safety and tolerability. The study will go on for 96 weeks with an ongoing extension phase. Today I'm presenting the primary endpoint from the week 48. The primary endpoint is the proportion of participants with plasma viral loads greater than 50 copies at week 48 by the snapshot intention to treat exposed analysis with a non-inferiority margin of 4%. Key secondary virologic endpoint is the proportion of participants who achieve virologic suppression at week 48. Additional secondary endpoints include safety and tolerability, incidence of confirmed virologic failure, viral resistance in participants experiencing virologic failure, as well as participant treatment preference. Here are the baseline characteristics for the cohort of the intention to treat exposed population. 1,045 individuals were randomized, and you can see good balance between the two groups. 63% of people had no prior exposure to cabotegravir plus rilpivirine. The median age was 42. Good representation of female sex at birth, as well as Black or African American race. 20% of individuals had a BMI greater than 30, and the median CD4 count was 661. If we look at the primary and secondary virologic endpoints, virologic non-response was seen in 1.7 and 1% of participants in the Q8 and Q4 week arm respectively, with the adjusted treatment difference being 0.8%, the 95% confidence interval crossing the null value but not crossing the 4% non-inferiority margin, and demonstrating that Q8 week dosing strategy is non-inferior to Q4 week. If we look at the secondary virologic success endpoint, you can see 94.3 and 93.5% of individuals in the Q8 and Q4 week arm respectively achieve virologic suppression with an adjusted treatment difference of 0.8, the 95% confidence interval crossing the null value but not reaching the 10% non-inferiority margin, and thus once again demonstrating that Q8 week is non-inferior to Q4 week at the week 48. If we look tabularly at these virologic endpoints, we can look closer at the individuals who failed to maintain a viral load less than 50 at week 48, and you can see the reasons listed here in this table. Furthermore, there were 21 and 29 individuals who had no virologic data, either because they discontinued for AE or death or discontinued for other reason. There were no clear patterns for these other discontinuations. Of note, discontinuations for AEs. There were a number of individuals who had injection site reasons for discontinuations. And then regarding other reasons, most commonly withdrawal by the participant or the investigator decision. If we look at the individuals with confirmed virologic failure, there were 10 individuals who met the threshold for confirmed virologic failure, 8 in the Q8 week arm and 2 in the Q4 week arm. This table demonstrates resistance that was detected at time of virologic failure. For rilpivirine resistance-associated mutations, 6 of 8 in the Q8 week arm and 1 of 2 in the Q4 week arm had evidence of mutations. For the integrase resistance-associated mutations, 5 of 8 in the Q8 week arm and 2 of 2 in the Q4 week arm had evidence of an integrase resistance-associated mutation. In a post-hoc analysis, we looked at PBMCs from baseline to look for archived resistance using HIV-1 DNA testing. The 2 individuals in the Q4 week arm had no evidence of archived resistance, while 5 of 8 from the Q8 week arm had evidence of pre-existing major rilpivirine mutations, and 1 of 8 had evidence of a pre-existing integrase mutation. There was no clear pattern in terms of the subtype or the country of origin for people who experienced virologic failure. 9 of 10 individuals resuppressed on fully active oral antiretroviral therapy. 1 person did not due to noncompliance to a PI-based therapy. All individuals retained phenotypic sensitivity to dolutegravir. There's going to be an ongoing analysis to look at the factors contributing to confirmed virologic failure, including baseline archived resistance as well as drug concentrations. The PBNC archived resistance for HIV DNA analysis is underway across the phase 3 program. In terms of safety and tolerability, here we have the AEs excluding injection site reactions. You can see 77% and 84% experience any AE. In terms of grade 3 or higher drug-related AEs, these were rare, with only 4 and 5% of individuals experienced those in the Q8 and Q4 week arms, respectively. And 5 and 8 individuals had a drug-related AE leading to withdrawal. In terms of serious adverse events, were very infrequently, and drug-related SAEs occurred in only 3 participants overall. Pre-syncope and acute pancreatitis in the Q8 week arm, and allergic reaction in the Q4 week arm. There was 1 fatal SAE due to sepsis in the Q8 week arm that was not related to study drug. Overall, 96% of the drug-related AEs were grade 1 or 2, and as noted, drug-related AEs leading to withdrawal occurred in only 5 and 8 participants respectively. Next, if we look at the injection site reactions, you can see the graph on the left highlights the injection site reactions. Day 1 reflects those individuals who were already on injectable therapy at the time of transition. And what you can see is at the early time points, week 4 and week 8, injection site reactions are most common, and then reduced to a background rate of approximately 20% over the course of the study. There were over 24,000 injections administered in total, with the majority of people not experiencing injection site reaction events with these injections. Grade 3 or higher injection site reactions were rare, occurring in only 43 and 48 individuals overall. The majority of injection site reactions were pain, and then much less frequently nodule and discomfort. People withdrew due to injection-related reactions in 6 and 11 participants overall in the Q8 and Q4 week arm, respectively. The majority of the injection site reactions, 98%, were grade 1 or 2, with a median duration of 3 days. And a very low number, less than 2% of participants, discontinued due to injection-related reactions. If we look at participant preference, individuals who are on oral therapy and enrolled into the Q8 week arm, 98% preferred Q8 week dosing to oral therapy. And if we look at individuals who had previously been exposed to the Q4 week arm, 94% preferred Q8, 3% preferred Q4 week dosing, and 2% preferred oral dosing. In summary, Q8 week dosing of cabotegravir plus rilpivirine long-acting was highly efficacious and non-inferior to Q4 week dosing, with very low rates of virologic non-response in both groups, and virologic suppression maintained in 94% of individuals in the study. The rate of confirmed virologic failure was low overall at 1%. Cabotegravir and rilpivirine were well tolerated with comparable safety profiles between the two arms. Injection site reactions were mostly grade 1 or 2 with a median duration of 3 days. 98% of participants preferred Q8-week dosing in treatment over the oral therapy, and Q8-week dosing was preferred by 94% of participants with prior Q4-week experience. In summary, cabotegravir plus rilpivirine long-acting, dosed every 2 months, is an innovative and effective treatment for the maintenance of virologic suppression in people living with HIV. I'd like to thank everyone who's contributed to the success of audD2a, and particularly thank Joe Polly for his help. I'd like to thank all the study participants and their families who volunteered their time and energy for the study, and also thank the audD2a clinical investigators and their staff for their efforts on this study. Thank you for your attention.
Thank you, Dr. Overton, for an outstanding presentation that's been highly awaited. You can send your questions to Dr. Overton at the email address that's on the slide, or post them to virtual CROI Q&A on Twitter. If you've been thinking about joining Twitter, now might be a good time. There's a lot of activity around CROI this year. So now I'm going to turn it over to Dr. Sag to introduce the next presentation.
Great, thank you, Judy. Our next presentation comes from Malawi. It's a prospective enhanced monitoring of dolutegravir-based first-line therapy in Malawi, presented to us by Elvis Timfak, and he's at the Medicine Without Borders site in Blantyre, Malawi. His disclosures are as follows: nothing to disclose. And so we'll turn it over to Elvis at this time.
Good morning, everyone. Since July 2018, WHO recommends a dolutegravir-based regimen as first-line ART. With the availability of fixed-dose tenofovir, lamivudine, and dolutegravir, there is wide-scale rollout of TLD in low and middle-income settings. With this rollout comes some key questions. Does TLD work for patients with 3TC and TDF resistance? At what rate will DTG resistance emerge? Which algorithm is best to identify treatment failure on TLD? In Malawi, since January 2019, there has been national rollout of TLD as first line for patients initiating ART, as well as those on NNRTI-based regimen transitioning to TLD. This transition was blind because viral load testing was not a prerequisite for transitioning in a setting where viral load testing is in scale-up and there is limited access to resistance genotyping. As such, there was need for a prospective study for the post-marketing surveillance of this new ARC policy change. MSF, in collaboration with the Ministry of Health, designed a study entitled Enhanced Monitoring of Efavirenz to Dolutegravir Transition. This study was carried out in the Chiradulu district in the southern part of Malawi, which has a high HIV prevalence with about 34,000 patients on ART managed in 11 health centers in the decentralized HIV program supported by MSF since 1998. In this district, viral load monitoring was introduced using a stepwise approach from mid-2013. The primary objective of this study was to describe virological response on TLD first line up to 18 months among patients transitioned from NNRTI to TLD, referred to as transitioners, as well as among patients initiating ART on TLD, with specific focus on patients non-suppressed at TLD start with or without underlying NRTI resistance. The study design was a descriptive prospective cohort carried out in 3 of the 11 health facilities in Chiradulu on a target population of 2,300 transitioners and up to 115 initiators. Inclusion criteria were those of the Malawian HIV guidelines for males above 20 years and females above 45 years, because at the time there was concern about the association between dolutegravir and neural tube disorder in women exposed to dolutegravir before pregnancy or during pregnancy. As such, pregnant women or those planning pregnancy were excluded. Patients with clinical signs of treatment failure, as well as those with a recent viral load above 1,000 copy in the past 6 months, were excluded. At study inclusion, blood was collected from all participants and plasma was prepared and stored for a retrospective plasma viral load testing, and in case viral load was above 100 copies, a resistance genotyping was done on this stored plasma. Participants were seen every 3 months, and during each visit, there was a clinical examination as well as neurotoxicity symptom inquiry. At milestone visits, plasma viral load was monitored at month 3, month 6, month 12, and at month 18. Given that the study is currently going on, we are going to share with you our initial findings at month 6 follow-up. Between January and May 2019, 1,928 participants were included, among whom 1,893 who were transitioners who had been on ART for a median of 7.7 years, 96% of whom were on a TDF-3TC based regimen. According to the results of the retrospective baseline viral loads, only 5.3% of transitioners had a detectable viral load, majority of whom were eligible for the pre-TLD drug resistance testing. Overall in the cohort, among the few initiators who were included, 80% suppressed by month 3, and by month 6, close to 90% had suppressed their viral load. Looking at the transitioners, there was already an excellent suppression on an NNRTI-based regimen before TLD, and this high suppression was maintained at month 3 and at month 6 on a TLD-based regimen. With focus on the transitioners, we will now discuss virological failure at month 6 follow-up on TLD. Among the transitioners in our cohort, where the majority were already suppressed at baseline, very few became detectable at month 3 and at month 6, around 1%. Among the few transitioners who were detectable at baseline, more than 85% suppressed at month 3 and at month 6. However, there was a higher rate of detectable viral load in this subgroup as compared to those who were baseline suppressed. In this study, at milestone visit, participants who had a viral load above 50 copies were considered as suspect virological failure, to whom adherence counseling was done and a repeat viral load done after 3 months. At repeat viral load above 500 copies, the participant was considered as a confirmed virological failure and a dried blood spot was collected for drug resistance. As such, at month 3, there was a low prevalence of suspect failure at 1.7%, only 16.7% of whom were confirmed failure at month 6. Considering this number on the overall tested at month 3 and at month 6, the prevalence of M6 confirmed virological failure was 0.3%. However, looking at the subgroup who were detectable at baseline, the prevalence of confirmed failure was 3.2%, and this represented about 10 times the prevalence among those who were baseline suppressed. Focusing on the characteristics of the 5 transitioners with confirmed failure, at baseline on the left side of the slide, we see that 3 out of 5 had high viral load at baseline, 2 of whom had TDF and 3TC resistance, and the other 2 were not detectable at baseline. At month 6, on the right side of the slide, we see that 2 out of 5 had DTG resistance at month 6, 1 with TDF-3TC resistance, and the other without. The other 3 patients who had virological failure at month 6 had no TLD resistance. This translates to a prevalence of confirmed failure with DTG resistance in the whole cohort of 0.1%, and 2.1% in the small subgroup with baseline detectable viral loads. As concerns safety, we had 2 cases of severe psychosis leading to TLD discontinuation before month 3. This psychosis was considered related to the treatment because there was symptom reversal after the participants were switched back to an NNRTI-based regimen. In summary, in a cohort well suppressed on NNRTI first line, overall high suppression was maintained at M3 and at M6 upon transitioning to TLD. Among the few with baseline detectable viral load, majority suppressed at M3 and at M6 on TLD. However, there was a higher prevalence of suspect failure in this group as compared to those who were baseline suppressed. Globally, there were few transitioners with confirmed virological failure at month 6, but of potential concern, 2 of these confirmed failures had DTG resistance, with the resistance-associated mutations being those commonly known among INSTI-naive patients. With our findings at month 6, longer follow-up is needed to better describe virological failure on TLD and incidence of DTG resistance. Follow-up is ongoing, and analysis at month 18 would integrate baseline resistance data as well as penetration measures at failure. There is also need to initiate active DTG surveillance in real-life prescription. At this point, please kindly permit me express our acknowledgement, firstly, to the study participants, the EMET team on the field, as well as all our international collaborators who are constantly working hard to make sure that the study is a success. Thank you very much for your attention.
Thanks so much, Dr. Timfak. And as everyone is looking at the screen and hopefully writing down the email address, normally you would have in your backpack a book that would allow you to write things down. So hopefully you'll have a piece of paper or some way to record this. And if you have any questions, please email Elvis. I'm sure he'd be happy to have a discussion with you. And I think we'll obviously be doing that for all the talks throughout the meeting. Judy?
Thanks, Mike. Okay, our next presentation is randomized switch to bictegravir FTAF in African American adults with HIV, to be presented by Dr. Debbie Hagans from the Chatham Care Center in Savannah, Georgia. Her disclosures are shown on the slide. Turn it over to Dr. Hagans.
Good morning, and thank you to the CROI planners and to Gilead for the opportunity to present this data on randomized switch to BF-TAF in African American adults with HIV. These are my disclosures. We want to express our sincerest gratitude to all the study participants and their families, to the study investigators and their teams, the Study Advisory Committee, and the North American HIV Research Community Advisory Group. This study was funded by Gilead Sciences. Black Americans have had the highest rates of HIV and AIDS in the U.S. for at least the past 2 decades, but remain underrepresented in clinical HIV studies. The single-tablet regimen of bictegravir, emtricitabine, and tenofovir alafenamide, BF-TAF, is a guidelines-recommended regimen both in the U.S. and in Europe, with demonstrated safety, efficacy, and a high barrier to resistance. In the international phase 3 naive studies of BF-TAF, 33% of participants identified as Black or of African descent. We evaluate the efficacy and safety of switching to BF-TAF compared to remaining on your current baseline regimen in virologically suppressed individuals who self-identified as African American or Black. The BRAVE 2020 study design is a U.S.-based phase 3 randomized, open-label, multi-center, active-controlled switch study in which adults living with HIV who were on 2 nukes plus a third agent had viral suppression of less than 50 copies for at least 6 months and had eGFRs of at least 50 or higher mL per minute and self-identified as African American, Black, or mixed race to include Black. They were randomized 2-to-1 to switch to BF-TAF or remain on their current regimen. Though this is a 48-week study, the primary endpoint was a 24-week FDA snapshot analysis of proportion of individuals who had confirmed virologic rebound of at least 50 or more copies. The secondary endpoints were proportion of individuals who maintained viral suppression at less than 50 copies and change in CD4 count from baseline. When we look at this AIDS-View map of Black individuals living with HIV in the U.S. in 2016, we see that the heaviest concentrations are in the southeastern United States, in the northeastern United States, and then there are pockets in the western region. The 82 sites that enrolled participants were deliberately selected to reflect the prevalence of Black Americans living with HIV. 558 individuals were screened and 496 were randomized. Through week 24, there were very small numbers of discontinuations, with 97% and 99% remaining on treatment in BF-TAF and the comparator arm respectively. When we're looking at baseline characteristics, the median age was 49 years with a range of 18 to 79 years of age. 1/3 of participants were female, and our study community advisors encouraged us to engage African Americans across a broad range. You see that 2% of the BF-TAF participants were trans women and 4% in the comparator arm, and those who identified as gay or bisexual were at 49% and 41% respectively, with the remainder of participants having a sexual orientation identified as heterosexual female and heterosexual male. Ethnicity of Hispanic or Latinx was at 5% and 3% respectively. In both treatment arms, median CD4 count and median EGFR was good. Median weight was at 88 and 89 kilograms, and those who had hepatitis B were allowed to participate in this study. Baseline nuke backbone primarily was FTAF at 2/3 in each study arm, at FTDF at about 20% in both arms, and 1 in 7 and 1 in 8 were on abacavir 3TC. The baseline third agent was more varied, which certainly reflects clinical practice, but 58 and 59% of individuals were in an integrase. Another approximately 30% were on a non-nucleoside, and 5 and 7% were on boosted darunavir. When we look at other and look at the footnote at the bottom of the slide, these other regimens were not present at 5% in the participants. Let's look at the ARB resistance according to historical data. PR resistance was 11 and 15% respectively. Non-nucleoside resistance was the most common at 20% approximately in both treatment arms, but we'll look more at the nucleoside resistance at 13% in BF-TAF and 60%, and then the subset who had the M184V and I at 9% in BF-TAF and 12% in the comparator arm. We look to the left of the slide at the 24-week analysis, and note that 2 study participants had historical resistance to integrases who were excluded from the primary analysis. Both have been randomized to BF-TAF and maintained viral suppression of less than 50 copies through week 24. We see that less than 1% had virologic confirmed rebound at greater than 50 copies, and that small numbers continue with 3% that had no virologic data. But the vast majority of participants maintained viral suppression. To the right of the slide, we see that BF-TAF met the criteria for non-inferiority, and change in CD4 count from baseline was not significant in either treatment arm. We look at the percent of participants who maintained viral suppression who had underlying nuke resistance. In the far left, we see that in the BF-TAF arm, 98% maintained viral suppression compared to the 96% who did not have any nuke resistance. And when we look to the right of those who had the M184V or I, we see again that viral suppression was maintained in both treatment arms. 1 participant in each group met the criteria for resistance testing through week 24, but there was no treatment emergent resistance in either treatment arm. For more details on resistance, please see Andrietta and others at poster 509. Adverse events of all grades occurred in very small numbers in both treatment arms, with the most common being URIs and joint pain. What is important is that there were no laboratory safety signals that emerged, and the hyperglycemia and glycosuria that occurred in 3 or 4 grade were in individuals known to have underlying diabetes. AEs that led to discontinuation of the study drug were again small, a total of 6. Most of them occurred in the first week, with one occurring at day 28. And this one participant who had acute kidney injury was diagnosed with prostate cancer that led to urinary obstruction. And there were no treatment emergent deaths through week 24. The changes in lipids from baseline, they were actually small reductions in both treatment arms. The cholesterol to HDL ratio, which we see on the right, was neutral. And please note that 27% of individuals in BF-TAF and 22 in the comparator arm were already on lipid-lowering drugs, and that there was not a significant initiation of lipid-lowering drugs through week 24 in either treatment arm. Weight is a topic of discussion for individuals on integrase-based regimens. So we'll look here at the median change in weight from baseline in the BF-TAF arm versus those who continued on their current regimen. The 0.9 kilogram, or just under 2 pounds, versus the 0.2 kilograms seen in the comparator group, just under a half a pound. While these changes are not significant, this is a relatively small time frame, and we need to follow this out to see what trends may unfold. Please note that some people lost and some people gained. In conclusion, switching to BF-TAF was not inferior to remaining on your current regimen of 2 nukes and a third agent for Black Americans living with HIV. Less than 1% of those who switched to BF-TAF had confirmed virologic rebound of 50 copies or greater at week 24. Those with pre-existing nuke resistance maintained high virologic suppression rates. No treatment-emergent resistance was observed. Switching to BF-TAF was considered safe and well tolerated as adverse events were comparable between the 2 treatment arms. Less than 2% of participants had AEs that led to discontinuations in this open-label BF-TAF study. It was lipid neutral, and the change in weight from baseline was not significantly different between the 2 treatment arms at week 24, but we need to follow this out further to see what trends develop. BF-TAF is safe and effective for people switching from a variety of antiretroviral regimens, including those with pre-existing nuke resistance. Thank you.
Thank you very much, Dr. Higgins. That was an excellent presentation. Congratulations on completing the study. As you can see, the email address for sending questions is on the slide here. And I'll turn it back to Dr. Sag.
Yep. As you're writing down the email address, we're going to transition now to eradication. And you heard about this morning from Sharon Lewin, and at her same institution, Thomas Rasmussen is one of the investigators that she alluded to in her talk, and this is the presentation on the impact of anti-PD-1 and anti-CTLA-4 on the HIV reservoir in vivo. It's the AMC 095 study, and as I mentioned, it's going to be presented by Thomas Rasmussen of the University of Melbourne in Melbourne, Victoria, Australia. Thomas? His disclosures are here.
Well, first of all, thank you very much for the opportunity to present our findings here, which are from a clinical trial of anti-PD-1 and anti-CTLA-4 in people with HIV and cancer. And I have no conflicts to declare. Antibodies to PD-1 and CTLA-4 have demonstrated anti-cancer efficacy, and this is due to the potent activation of tumor-directed T-cell responses. Studies have also shown an enhanced therapeutic efficacy with combined blockade of both PD-1 and CTLA-4, but also increased toxicity. And there is also still limited knowledge of the use of these antibodies in people living with HIV. In addition, other studies have shown that HIV persists preferentially in cells that express CTLA-4 and PD-1 in both blood and lymph node tissue. So therefore, it is possible that antibodies against PD-1 and CTLA-4 could target HIV persistence on ART by reversing HIV latency and or boosting HIV-specific immunity. And there is some evidence to support this thinking. For example, it was recently shown that blocking PD-1 in vitro could enhance latency reversal, and at CROI last year there was also shown studies to support some degree of latency reversal in vivo. In addition, we previously reported a substantial increase in both cell-associated and plasma HIV RNA in an HIV-infected individual receiving anti-CTLA-4 for melanoma. So collectively, this raises the question of whether anti-PD-1 either alone or in combination with anti-CTLA-4 might reverse latency, and given the ability of the immune check antibodies to enhance T cell function, whether this might facilitate elimination of latently infected cells. And through a collaboration with the AIDS Malignancy Consortium, we've been able to address this question in the setting of an ongoing phase 1 clinical trial of anti-PD-1 and anti-CTLA-4 in people with HIV on ART with advanced malignancies. And whereas the overall objectives of this study is to assess safety and anti-tumor response rates, we've been able to embed a reservoir sub-study that specifically addresses what is then the impact of anti-PD-1 either alone or in combination with anti-CTLA-4 on HIV latency, on the frequency of latently infected cells, and on HIV-specific immunity. And what I'll be reporting here is the latency reversing effects and any impact on the reservoir of the immune checkpoint antibodies, whereas the ability of immune checkpoint blockade to enhance HIV-specific T cell function is something that we are still investigating in the lab. So this was a non-randomized phase 1 multi-site clinical trial with enrollment ongoing at 31 sites, primarily in the US, but also a single site in Australia. And although the study design is a little complex, for this purpose it can be simplified to the diagram shown here. So the study enrolls people with HIV on ART with cancer and is doing this across 2 stratum based on CD4 counts above 200 or CD4 counts between 1 and 200. Study participants are then assigned to receive either single-agent anti-PD-1 in the form of nivolumab 3 milligrams per kilo or a flat dose of 240 milligram given every 2 weeks. Other participants are assigned to receive combination anti-PD-1 and anti-CTLA-4, and again this is nivolumab given every 2 weeks and ipilimumab given at a 1 milligram per kilo dose every 6 weeks. Participants can receive treatment for up to 92 weeks, but this is obviously dependent on disease progression, toxicity, and anti-tumor response rates. For the reservoir sub-study, we then collected plasma and PBMCs at baseline and then at days -1, +1, and +7 relative to the infusion of immune checkpoint antibodies. And this was done at cycles 1, 4, 7, 10, and so forth to correspond to the cycles that included both anti-PD-1 and anti-CTLA-4 for those on a combination regimen. And as you might imagine, in a study like this, there is a relatively high dropout rate due to death, toxicity, or disease progression. We therefore primarily have samples available in cycles 1 and 4 and have focused our analysis in these 2 cycles. In addition, a subset of participants also provided a large volume blood draw both at baseline and usually at the end of study therapy that enabled us also to perform quantitative viral outgrowth assay in a subset of participants. We then assessed the effects on reversing latency by quantifying the level of cell-associated unspliced HIV RNA in total CD4 T cells using real-time PCR, and we also quantified the levels of plasma HIV RNA using replicate testing on the Aptima HIV1 quant assay. To investigate effects on the reservoir, we quantified the level of cell-associated total HIV DNA in total CD4 T cells, and also performed quantitative viral outgrowth assay in a subset of participants that provided a large volume blood draw. In the end, we've been able to enroll samples from 40 participants in the sub-study, and their baseline characteristics are shown here. And I'll just briefly highlight that these were predominantly male, and they had a median CD4 T cell count at baseline of 315. And also that of the 40 participants, 33 received anti-PD-1 alone, and 7 received combination therapy. In terms of malignancies, the study included samples from 15 people with KS, but otherwise a fairly heterogeneous group of malignant diagnoses represented in the study. To initially assess any latency reversing effect of the immune checkpoint antibodies, we then quantified the level of cell-associated HIV RNA in total CD4 T cells, as shown here. And at the top of the graph, you can see the time points for infusion of the immune checkpoint antibodies in cycle 1 and 4. And also note that blue dots represent participants that received anti-PD-1 alone, and red dots those on combination therapy. And as you can see, when we analyzed the entire cohort, overall we detected no significant change in the level of cell-associated unspliced HIV RNA during immune checkpoint blockade. Similarly, we detected no significant changes in 33 participants that received anti-PD-1 alone, but in 7 participants that received both anti-PD-1 and anti-CTLA-4, we did detect a significant increase that was present at the first time point, roughly 24 hours after receiving the first dose of both antibodies. And this increase may not be particularly clear on this graph, but if you plot the individual data points, you can see that the increase is actually quite consistent, is happening for 6 of the 7 participants, and corresponds to a median fold change of 1.44. The increase was actually even further marked at the time point 24 hours after the dose in cycle 4, where it corresponded to a fold change of almost 2.5, but with only 4 samples available at this time point, this did not reach statistical significance. Next, we then investigated whether immune checkpoint antibodies might also increase the level of plasma HIV RNA. And as mentioned, this was done using replicate testing with the Aptima HIV1 quant assay, and we only performed this assay in samples from cycle 1. And doing this, we were able to detect a minor but statistically significant increase at day 7 after starting immune checkpoint therapy, and this corresponded to a 17% increase in plasma HIV RNA. We obviously also analyzed changes within those receiving anti-PD-1 alone or combination therapy, but did not detect any significant changes within these groups on their own. Then, to address whether immune checkpoint blockade might also have an impact on the frequency of latently infected cells, we initially quantified the frequency of cells carrying total HIV DNA using real-time PCR, and these data are shown here. And although we detected a moderate decrease of 45% at the first time point, 24 hours after the first dose, this decrease was not sustained beyond that time point, and we speculate that it may simply represent cellular redistribution. When we applied a generalized negative binomial regression, this also identified a significant decrease over the entirety of the first 4 cycles, but this was with a very low effect size, and we tend to think that this finding do not carry any real biological and at least clinical meaning. We then performed a quantitative viral outgrowth assay in participants that provided the additional consent for a large volume blood draw both at baseline and during immune checkpoint blockade to estimate the frequency of cells carrying replication-competent HIV. This was possible to perform in 10 individuals, and in those 10 we did not detect any significant change in the frequency of cells with replication-competent HIV expressed as infectious units per million. Similarly, in 8 participants on anti-PD-1 alone, we did not see any significant change in IUPM, but in 2 of 2 individuals receiving combination therapy and with samples available for this assay, we did see a decrease in IUPM in both of these 2. So this brings me to my conclusion that this is really the first study that's been able to investigate effects of both anti-PD-1, either alone or in combination with anti-CTLA-4, on HIV latency and on the frequency of latently infected cells. And although the study provides some evidence for a latency reversing effect of combined blockade of both PD-1 and CTLA-4, the effects were relatively modest. Overall, we saw no change in the frequency of cells that contained replication-competent HIV, but we did note a decrease in 2 of 2 individuals receiving combination blockade and that had samples available to perform QOA. We fully acknowledge that current licensed antibodies to anti-PD-1 and anti-CTLA-4 have considerable toxicity, which is challenging their use in people that are doing well on ART. But we also argue that it is entirely possible that in the future we may see improved formulations with lower toxicity or a better understanding of toxicity or better strategies to mitigate toxicity. And we therefore believe that understanding these effects working through the immune checkpoints is still highly relevant to efforts of achieving HIV remission. And finally, I also wanted to emphasize that the principal effect of immune checkpoint blockade is to enhance T-cell function. This is the basis for their use in cancer, and this is something that we are still investigating with regard to their effect on enhancing HIV-specific T-cell function. And with that, I also wanted to acknowledge a great group of collaborators and investigators that have contributed to this study. I specifically would like to highlight Sharon Lewin, Steve Deeks, and Christine Durand, who have been major forces in driving this study forward and were key to setting it up at the beginning. I also want to acknowledge a very generous collaboration with the AIDS Malignancy Consortium and their investigators listed here. Also want to thank the study funders and obviously the participants in the AMC 095 study. Thank you.
Thanks very much, Thomas. That was a wonderful presentation, very cutting edge, and something that a lot of us have wanted to see for quite a while, so it's really exciting to see your results. Thomas's email is on the screen now. Again, you can jot it down, and if you have questions or comments, I'm sure he would love to hear from you and develop a correspondence about his presentation. Judy?
Thanks, Mike. Okay, our next presentation is a randomized trial of the impact of 3-BNC117 and romidepsin on the HIV reservoir by Dr. Ole Sograd from Denmark. Disclosures are shown on this slide and onto the presentation.
First of all, I'd like to thank the organizers for selecting this abstract for all presentations at CROI. The background for this study is the so-called shock and kill strategy to eliminate the latent reservoir. In this strategy, a latency reversing agent is administered to patients on long-term suppressive ART. This in turn turns on the transcription of integrated provirus in latently infected cells. These cells are subsequently eliminated either by viral cytopathic effects or immune effector mechanisms. Several clinical studies have found that different latency reversing agents can induce or enhance HV transcription, but so far there has been no overall reduction in the size of the latent reservoir. We decided to initiate a clinical trial called the Roadmap study, which aimed to investigate if the administration of broadly neutralizing antibodies prior to latency reversion could help eliminate latently infected cells. The primary objective of this study was to evaluate the effects of romidepsin plus 3-BNC117 or romidepsin alone on delaying or preventing viral rebound in HIV-infected individuals during an analytical treatment interruption. The primary endpoint was days to viral rebound during the HI, defined as plasma HVRNA above 200 copies per ml on 2 consecutive measurements, or days to reinitiation of ART in patients who started ART before viral rebound. 3-BNC117 is a broadly neutralizing antibody targeting the CD4 binding site on HV envelope. 3-BNC117 has been shown to suppress viremia in untreated individuals and to delay time to viral rebound in those that interrupt ART and are dosed with antibodies into the treatment interruption. Data also indicates that 3-BNC117 facilitates the elimination of HV-expressing cells. And finally, the safety and tolerability of this compound is well documented with no severe adverse side effects in more than 100 treated patients to date. Romidepsin is an HDAC inhibitor licensed in the US for the treatment of peripheral T-cell lymphomas and cutaneous T-cell lymphomas. It has been shown to increase extracellular RNA released from memory and resting CD4 T cells in patients ex vivo, and a pilot study done in 2014 showed that romidepsin induced HV transcription, leading to quantifiable viremia in 5 of 6 participants. In the study, participants received 2 cycles of romidepsin, where Group A received 1 dose of 3-PNC117 prior to romidepsin, both in the first and the second treatment cycle, whereas Group B only received romidepsin. The study population were similar in the 2 groups, with the romidepsin alone group having a slightly higher median age and having been on long-term ART for a median of 9.5 years compared to 5 years for the combination group.
Previous.
The most common adverse events were nausea followed by headache, fatigue, malaise, chills and vomiting. ECGs were obtained before romidepsin 1 hour into the infusion and at the end of infusion. 2 participants experienced QTc prolongation after romidepsin, which resolved spontaneously by 1 week post-infusion. No other clinical changes were observed in the ECGs. CD4 cell counts remained stable for both groups, from baseline to midway between 2 cycles to just prior to interrupting treatment, with no significant changes occurring at the 2 latter time points compared to baseline. The effect of romidepsin on HV transcription was analyzed by measuring cell-associated unspliced HV RNA in total CD4 T cells at multiple time points. During the first treatment cycles, minor increases in HV RNA was observed for both groups, and during the treatment cycles these changes were somewhat more pronounced. Compared to some of the previous clinical trials, the fold changes were not as pronounced, but it's important to note that this study did not include a post-infusion time point where the change in HV transcription following romidepsin administration appears to be most pronounced. The effect of romidepsin on plasma HV RNA was also investigated at multiple time points during treatment cycle 1 and treatment cycle 2. While multiple individuals in both groups had blips above the lower level of quantification, there were no consistent increases following romidepsin infusion in either of the 2 groups. Of note, 1 individual from the romidepsin group stopped taking ART during the romidepsin treatment and was excluded from this and further analysis. The effect of the interventions on the size of the HV reservoir was determined by measuring the total HV DNA content in isolated CD4 T cells. There were no significant changes from baseline to midway between the 2 cycles and just prior to interrupting treatment in either of the 2 groups. For patients in the combination group, we determined baseline sensitivity to 3-PNC117 by sequencing HV envelope from profile DNA at baseline and using these sequences to infer sensitivity according to 2 different protocols. The first protocol we applied was the so-called sensitivity protocol, which estimated that 6 of the 7 patients with envelope sequences were sensitive to 3-PNC117, whereas the escape protocol estimated that 6 of 7 patients were resistant to 3-PNC117. For 4 individuals, we had no sensitivity data as there was no amplification of HV envelope using 2 different primer sets. The primary endpoint of the study was time to viral rebound during the ATI. Shown here are the rebound curves for all individuals in the study. The combination arm that received both 3-BNC117 and romidepsin are shown in red, and the romidepsin arm is shown blue. As you can see, all patients rebounded, with one patient in the romidepsin group rebounding at week 12. It's important to stress that there were a minimum of 16 weeks from the last 3-BNC117 infusion to the start of the HVI, and therefore the serum concentrations of the antibodies at the start of HVI were not suppressive. Putting these data into a Kaplan-Meier plot shows that the romidepsin alone arm rebounded slightly later than the combination group, with a median of 28 days to reaching the primary endpoint compared to 17.5 days for the combination group. Log-rank test applied to these data gave a p-value of p.02. This difference was mainly driven by the late rebounding individual. This graph shows HV-specific CD8 T-cell immune responses by ICS for the two groups, with the combination group at the top. As you can see, there is no significant increases or decreases in HV-specific immunity from baseline to just pre-HI. To conclude, the two groups were comparable in their baseline characteristics, although the romidepsin alone group had been on ART almost twice as long as the combination group. We found modest latency reversal despite multiple romidepsin infusions. No evidence of a decline in the size of the viral reservoir for either groups. No clear delay in time to viral rebound. No induction of HV-specific immunity in the 3-BNC117 group. Also, we found no evidence of detrimental effects of romidepsin on HV-specific immunity. Thus, we conclude that latency reversal with the currently available LRAs and boosting of autologous HV-specific immunity may not be very effective in long-term suppressed patients. I would like to acknowledge first of all the study participants for their incredible efforts to participate in this very complicated trial. The collaborators at the Rockefeller University, in particular Marina Kasky and Michelle Nussenzweig, the people at my Department of Infectious Diseases in Aarhus, in particular Jesper Damsgaard Gunst and Marie Høst Pohus, and the people at Cologne University Hospital, in particular Gert Fettenhøjer, Florian Klein, and Henning Grüll. Finally, I would like to give a big thanks to AMFA for funding this trial.
Thank you very much for an excellent presentation. Once again, you see the email address for sending questions. And also, just make a comment that posters are available on the app, and they'll be available on the website subsequently. Okay, Dr. Sag, back to you.
Thanks, Judy. We're going to transition back to therapy at this point in time and go out to Los Angeles, California. Dr. Peter Ruan, who is going to present to us today on the safety and pharmacokinetics of GS9722 in HIV-negative participants and people with HIV. Dr. Ruan's disclosures are on the next slide, and you can see them here. And now we're going to transition.
Good morning, everybody. My name is Dr. Peter Ruan. I'd like to thank the conference organizers for the opportunity to present these two trials in elapovimab today. We extend our thanks to the participants, their partners and families, and all the GSUS 420, 3901, and 3902 investigators, and a special thanks to the study teams. And I'd like to offer my special thanks to my associate, Dr. Peter Wolf, research director Danny Ain, and team members Lucy Rennell, Nabila Hernandez, and Matthew Weissman, who worked so hard on this study at my site in Los Angeles. These studies were funded by Gilead Sciences. Elapovimab, which I will refer to going forward as EVM because it's a lot easier to say, is an effector-enhanced broadly neutralizing antibody that targets a V3 glycan motif of the HIV envelope protein. EVM is engineered from PGT121. PGT121 has demonstrated efficacy in combination with the TLR7 agonist vesatolimod for targeting and clearing the viral reservoir in HIV-infected primates. EVM has modifications intended to enhance Fc affinity for effector cells and improve serum half-life. It is being developed for use in an HIV cure regimen. The two trials I'm describing here today, EVM was administered as an IV infusion to HIV-negative participants. This was a phase 1A first-in-human trial in healthy volunteers, and a phase 1B study where it was administered to HIV-positive participants who are virologically suppressed on ART. This morning, I'll report to you the safety, tolerability, and the results of pharmacokinetics of EVM in the two populations studied. Elapovimab is similar to the broadly neutralizing antibody PGT121, but has undergone molecular engineering aimed to enhance effector function, optimize PK, reduce the risk of immunogenicity, and also make it easier to produce. For example, in studies previously presented, EVM proved more efficient than PGT121 in recruiting monocytes and NK cells for monocyte and NK cell-mediated cell kill. This is the design of the phase 1A study. Please note that it has a very similar design to the phase 1B study that I'll also review. This is a randomized, double-blind, placebo-controlled dose escalation study of EVM. The randomization was 6:2 drug to placebo. There were 49 participants. EVM was given in a single ascending dose, indicated here on the slide and throughout as SAD or SAD. And this was administered in one of three doses, 150, 500, or 1500 milligrams. There was also multiple ascending doses, indicated here on the slide as MAD, M-A-D. And in the MAD group, participants received 150, 500, and 1000 milligrams. And each of these doses was repeated twice at 2-week intervals for a total of 3 infusions. PK was performed following each dose. The primary objective of the study was safety, tolerability, and PK of EVM, with the secondary objective the development of anti-drug antibodies. Shown here are the baseline characteristics of the HIV-negative healthy volunteers for both the SAD and MAD groups with the corresponding doses of EVM received. Most participants were in their 30s, with the majority being male, and most participants were Hispanic or Latinx by ethnicity. This is the safety summary for both the SAD and MAD groups, including placebo. As might be expected with the administration of a human IgG antibody, these infusions were well tolerated with only few side effects. The only adverse event reported in more than one participant in either group was back pain, and this was reported in 2 participants in the 1000 milligram multiple dose group. There were 2 adverse events that led to premature discontinuation of the drug. One participant experienced a grade 2 AE of an infusion-related reaction at the start of the second infusion of EVM 1000 milligrams and was discontinued. One participant experienced a grade 3 SAE of thrombocytopenia following the first infusion of EVM 1000 milligrams. The onset of thrombocytopenia was day 12 after the infusion. The platelets nadered at day 14 and recovered to baseline by day 25 after the infusion. The patient was hospitalized for observation, but was asymptomatic throughout. No specific treatment was offered, and the patient was not redosed with EVM. Shown here are the key summary pharmacokinetic results from both the SAD and the MAD groups. This is a busy slide. Let me direct your attention first to the tables showing the numerical results for the pharmacokinetics of EVM according to the dose administered in each group, with key parameters shown in the left-hand column. These are AUC, Cmax, and relevant Cmin, expressed as numerical mean values along with the coefficient of variation. Also shown in the tables is the estimated median half-life in days, also with interquartile ranges. The figure over to the right is easier on the eye. Shown are the overlapping serum EVM curves from the MAD group with peaks following each of the ascending doses, 150, 500, and 1,000 milligrams, given on days 0, day 14, and again on day 28, with decay curves extending out to day 112. From both the tables and the figure, the following observations can be made. The pharmacokinetics of EVM are nicely linear and dose proportional over the evaluated range. The calculated serum half-life was about 26 days, supporting every other week dosing. 9 of 37 participants developed anti-drug antibodies, but the presence of anti-drug antibodies did not impact EVM PK in any participant. The stipulated horizontal line shown in the figure denotes the target Cmin of more than 50 micrograms per ml. 50 micrograms per ml is 3 times the IC95 used to determine in vitro breadth of EVM. This is an estimate of what level of EVM may be needed in a cure setting. Both the 500 milligram and the 1,000 milligram doses, administered every 2 weeks, maintain trough levels well above the 50 microgram per ml target level. The design of the phase 1b study in 32 ART-suppressed patients was very similar to the design of the phase 1a study, but with doses informed from the PK and other results in the phase 1a study. Here, patients were randomized to receive EVM in the SAD group as a single dose of either 150 milligrams or 500 milligrams. In the MAD group, patients received either 150 milligrams or 500 milligrams, given at 2-week intervals for a total of 5 infusions per patient. Secondary objectives in this study included development of anti-drug antibodies, also measurement of HIV RNA reservoir, and other immunological assessments that will not be presented today. Shown on this slide are the baseline characteristics of the HIV-positive participants in the phase 1b study. Compared to the phase 1a study, these participants were a little older, mostly male, all fully suppressed less than 50 copies, with good immunological recovery, and in general, good health. Administration of EVM in single and multiple doses of 150 and 500 milligrams was safe and well tolerated in this HIV-positive group. There was 1 grade 3 elevation of ALT in a participant in the MAD group that occurred at day 115. This was worked up but was otherwise unexplained and was judged related to the study drug by the investigator. It had resolved to grade 1 at the last study visit on day 225. In the placebo group, there was 1 SAE due to small bowel obstruction in a person who previously had small bowel obstruction. The pharmacokinetics and curves in this study with fully suppressed HIV-positive patients was very similar to those shown in the previous study of healthy human volunteers. This time in the figure to the right, you'll observe 5 peaks of EVM corresponding to each dose administered. EVM PK in these patients with fully suppressed HIV was also linear, dose proportional, and consistent with the results from healthy volunteers. As you can also see from the figure, EVM dose of 500 milligrams given every 2 weeks maintained levels well above the target of 50 micrograms per ml. 9 or 37% of the 24 participants developed ADA, but the presence of ADA did not impact EVM PK in any participant. Hello again. So in conclusion, EVM dosed up to 500 milligrams is generally safe and well tolerated in HIV-negative participants and virologically suppressed patients living with HIV, with similar single and multiple dose PK in the two populations. One HIV-negative participant developed grade 3 thrombocytopenia after a single 1,000 milligram dose which recovered to baseline. Just a little bit more information on that individual. There was an extensive workup pursued. Platelet antibodies were detected during the workup in the hospital, but subsequently serum binding of platelets whether in the presence of EVM or not could not be demonstrated. Anti-drug antibodies were not detected in this individual until day 58 after the dose administered. EVM PK was linear and dose proportional over the evaluated range. The estimated half-life of 26 days supports EVM dosing every other week, and these data support ongoing evaluation of EVM as part of a combination therapy for HIV cure. Thank you very much for your attention.
Thanks a lot, Peter. Especially liked your hello again at the end. That was very nice. This is how you can reach Peter at ruanmedical.com. I'm sure he'd enjoy interacting with you via email. Going to continue with the program. Turn it back to Judy.
Okay, thanks, Mike. The next presentation is Safety and Analytic Treatment Interruption Outcomes of Vesatolimod in HIV Controllers. That will be delivered by Devi Sengupta from Gilead Sciences. Disclosures are shown on this slide. I'll turn it over to the presentation.
Good morning. On behalf of the study team, I look forward to sharing data from our early phase study of Vesatolimod in HIV controllers that we believe sets a promising path for combination regimens. This was a large team effort across several institutions, and most of all, we'd like to thank the participants for their dedication to this intensive trial. Preclinical studies with an agonist of the innate immune sensor TLR7 suggest that it could play a role in the enhancement of CD8-mediated viral control when combined with a therapeutic vaccine, likely through multiple effects. We hypothesized that people living with HIV who have a partially effective CD8 T-cell response prior to ART, viremic controllers, may benefit from immune modulation with the investigational TLR7 agonist Vesatolimod, leading to improved viral control post-ART, similar to the NHP results. We report the first findings of a phase 1b placebo-controlled ATI study of Vesatolimod in controllers on ART. Individuals with a historical set point between 50 and 5,000 copies, who were subsequently suppressed for at least 6 months on ART, were randomized 2-to-1 to receive 10 doses of Vesatolimod or placebo. The dose level was escalated within participants, with the majority of individuals receiving 6 milligrams. After completing dosing, they stopped ART in a 24-week analytical treatment interruption to allow assessment of viral rebound. The primary objective was safety, and we also assessed immunologic markers such as interferon-stimulated genes, cytokines, cell activation, and HIV-specific T-cell responses, as well as measures of the HIV reservoir. In the ATI, we compared time to rebound and set point. ART was restarted if viral loads were sustained above 10,000 copies for more than 4 weeks. And in individuals who maintained low viral set points after the first 24 weeks of the ATI, an optional extension was offered. We felt that the placebo group was important to include in this study since the effect of ART itself has not been previously reported in a viremic controller population. We enrolled participants in their 40s and 50s and were pleased to be able to include 4 women and a diverse group of individuals. At study entry, the viral loads were suppressed with well-preserved CD4 counts. And individuals had been on ART for about 3 years, with pre-ART viral loads of about 1,000 copies. Similar to our previous dose escalation study, Vesatolimod was generally well tolerated, with no discontinuations due to adverse events. Some participants experienced the expected mild flu-like symptoms. However, these did not occur with each dose. After the study drug dosing period, during which we did not detect consistent plasmaviral blips, participants entered the ATI and were monitored weekly for at least 24 weeks or until they met ART restart criteria. Post-ART viral rebound has not been systematically studied in controllers, and we noted some interesting patterns overall across the participants. I'll be presenting the between-group differences on the next slide. Rebound to greater than 50 copies occurred between 2 to 15 weeks, with a median of 4 weeks. Peak viral loads of about 10,000 copies were reached, with some individuals going higher than that, but most came down within a few weeks to low set points. This suggests that the immune responses needed to contain replication to low levels may need to be primed by relatively long periods of viremia. which could have implications for ATI study designs. We're planning to look at these kinetics more closely through mathematical modeling. In the 23 participants who proceeded to the ATI, these are the results for time to viral rebound above 200 copies. We noted a small but statistically significant longer time to rebound in the group receiving vesitolimod compared to placebo. Within the vesitolimod group, there was also a trend for a reduction in set point compared to pre-ART values. And the median time to restarting antiretroviral therapy was 47 weeks for the vesitolimod group and 28 weeks for placebo. There was one participant who did not rebound until 31 weeks. This individual did not have any remarkable baseline characteristics, received the 6 mg dose level of vesitolimod, and maintained low viral loads below 200 copies for about 1 year, ending the study off ART, having achieved about a 1 log decrease in set point. We view these findings from this controlled trial as an encouraging step towards building combination therapies and are working to understand the mechanisms of these observations further. One of our questions was whether vesitolimod had any effect on the latent HIV reservoir. We did not see an impact on the total DNA in either group. However, the intact proviral DNA assay detected decreases from baseline in the vesitolimod group. This assay is designed to measure sequences that represent the replication-competent reservoir, and we look forward to larger studies to understand the clinical significance of these changes. Other measures such as inducible virus production, cell-associated RNA, and single-copy assay are pending. Turning now to the immunologic outcomes, first, we observed evidence of target engagement by dose-dependent changes in antiviral ISG expression, the most sensitive marker of TLR7 activity. Cytokines and chemokines such as IL-1RA, IP10, and ITAC, which regulate immune cell trafficking to the gut, also increased with each dose administration, while plasma interferon alpha was minimally detected, which is consistent with the tolerability of the drug in this study. Vesitolimod activated CD4 and CD8 T cells, as measured by Ki67 and CD38 expression, respectively, particularly in the transitional and effector memory subsets. We also noted that participants with delayed viral rebound tended to have higher activation in the CD8 T cell compartment. Our previous study also showed NK cell activation, and we're currently analyzing those data for this study. These measurements in the peripheral blood may reflect a stronger signal in local sites, such as the gut, where vesitolimod is designed to be the most active, but we did not obtain those samples in this study. Finally, analyses of the HIV-specific CD8 T cell response are ongoing, but I'd like to share these preliminary results from intracellular cytokine experiments in which PBMCs were stimulated with GAG, POL, envelope, and NEF peptide pools, and the frequency of cells expressing the cytokines listed here was measured. These are the changes in total magnitude from baseline to pre-ATI and during an ATI remission time point across the vesitolimod group. We're also analyzing changes in polyfunctionality, as shown in this individual example of a participant who had an increase in polyfunctional CD8 responses to HIV antigens. While we don't see dramatic effects in any one peptide pool, when we look at the aggregate response, the majority of the vesitolimod group appears to have increases in response magnitude and/or polyfunctionality, and we look forward to reporting detailed results in the future. In conclusion, in HIV controllers compared to placebo, multiple vesitolimod doses up to 8 milligrams were well tolerated, did not impact the total HIV DNA, but were associated with decreases in intact DNA as estimated by the audD2a assay. We also noted modestly increased time to viral rebound after the ATI and decreased viral set points. These effects are potentially due to an augmented antiviral immune response, as suggested by the induction of ISGs, cytokines, activation of immune cells, and increases in polyfunctional HIV-specific responses in a subset of participants. We believe that trials evaluating the efficacy of vesitolimod in combination with CD8 vaccines and monoclonal antibodies are warranted and are ongoing. Since there isn't an opportunity for live Q&A in this session, please feel free to reach out by email with any questions. And thank you very much for listening.
Thank you. You'll see the email on this slide. I've been reminded that some pharmaceutical company policy precludes commenting on social media, so please do use the email, and we all regret the inability to have an interactive Q&A. Because we don't have the presenters online, we aren't able to ask questions to them, so that's why we're not doing that. All right, back to you, Dr. Sag.
So our presentation for this session is really exciting. It's using an adeno-associated virus to do transfer and production of antibody, and it's going to be presented by Joseph Casaza, who's at the National Institutes of Health Vaccine Research Center. The title of the presentation is Durable HIV Antibody Production in Humans After AAV8-Mediated Gene Transfer. And the disclosures are on the next slide. And we'll now start the presentation.
In 2013, David Baltimore suggested that the VRC sidestep the difficulty of designing an effective HIV immunogen by using an adeno-associated viral vector to transfer genetic material coding for the production of broadly neutralizing antibodies directly to humans. Adeno-associated viral vectors have a number of characteristics that make this a reasonable possibility. They have an established safety record in humans. The transgene persists in the nucleus as an episomal DNA, which is stable in postmitotic cells. Capsids of multiple serotypes are available for use, some without significant pre-existing human seropositivity. Importantly, with adeno-associated viral vectors, transduction occurs in vivo, which allows adeno-associated viral vectors to be given by IM injection or by IV infusion. There are limitations, though. Adeno-associated viral vectors can at most carry between 4.7 to 5 kilobase coding cassettes, which, while not large, is adequate for many purposes. The genetic structure of adeno-associated virus is straightforward. They are single-stranded DNA viruses which code for 2 genes. These genes are flanked by 2 inverted terminal repeats. The inverted terminal repeats contain a 145 base palindromic sequence, which is mirrored on each side of the rep and cap genes. The inverted terminal repeats are required for DNA packaging and to form the sticky ends needed to produce episomal DNA. The simplicity of adeno-associated virus's genetic structure make it an ideal vehicle for gene transfer. Although the inverted terminal repeats are essential for vector construction, the rep and cap genes are not. They can be deleted and replaced with the desired coding cassette. Rather than producing 2 coding sequences under the control of 2 different promoters and 2 different poly-A sequences as some have done, Dr. Balazs and Baltimore designed a cassette containing a single promoter followed by a heavy chain IgG coding sequence, an F2a ribosomal jump sequence, a light chain IgG sequence, and then a single poly-A sequence. The F2a sequence allows the ribosome to jump from the IgG heavy chain directly to the IgG light chain, thus producing 2 separate proteins from 1 mRNA sequence. This results in the production of nearly equal amounts of IgG heavy and light chains and a considerable savings in genetic real estate. Doctors Balazs and Baltimore used these savings to enhance the transcription of vector DNA by introducing a woodchuck hepatitis virus post-translational regulatory element. In 2013, the most potent CD4 binding site antibody available to the VRC was VRC07. Like VRC01, VRC07's CDRH2 loop binds to the CD4 binding site of GP120. VRC07 also has a 4-amino acid insert into the CDRH3 loop, which allows the antibody to further stabilize GP120 binding by binding the inner domain of GP120. This results in a twofold lower IC80 with most viral species and an increased breadth of viral neutralization. Despite these improvements in function, VRC07 is similar to VRC01 in structure. Based on amino acid sequences, the light chain of VRC07 is identical to VRC07. The heavy chain is 85% identical. This similarity to VRC01 suggests that, like VRC01, which has been given to over 3,000 volunteers without evidence of an audA response, VRC07 should be unlikely to induce a significant audA response. Equally important, the high affinity and specificity of the anti-idiotype antibody 5C9 to VRC07 has allowed the development of high-sensitivity VRC07 assays. After extensive preclinical studies, VRC603 opened in January of 2018. VRC603 is an open-label, Phase 1 dose escalation study designed to study the effects of the administration of AAV8 VRC07 by IM injection to humans. The major goals of this trial are: 1. To evaluate the safety and tolerability of AAV8 VRC07 given by IM injection to HIV-infected individuals on effective antiretroviral therapy. 2. To characterize the pharmacokinetics of AAV8 VRC07-induced VRC07 production. And 3. To follow the effects of gene transfer of VRC07 to humans by AAV8 VRC07 over a 5-year period. VRC603 has 3 dose groups: a low dose group, which will receive 5 times 10 to the 10th vector genomes per kilogram, an intermediate dose group, which will receive 5 times 10 to the 11th vector genomes per kilogram, and a high dose group, which will receive 2.5 times 10 to the 12th vector genomes per kilogram. Because the concentration that AAV vectors can be produced is limited, individuals in the high-dose group are given multiple injections, rotating from the right shoulder to the left shoulder to the right quadriceps to the left quadriceps, and then repeating that cycle until the appropriate dose is administered. To date, we have enrolled 8 HIV-infected individuals in VRC603, 6 men and 2 women. 5 of the 8 volunteers are African Americans. All but 1 individual had a viral load of less than 20 at enrollment. Median CD4 count was 528 cells per microliter. Median age was 52 years of age. To date, 3 subjects have been enrolled in the low dose group, each receiving product in 2 IM injections. These individuals have been followed for up to 2 years and 1 month. 2 of 3 subjects have been enrolled in the intermediate dose group, each receiving product in 2 IM injections. These individuals have been followed for up to 1 year and 10 months. 3 of 5 individuals have been enrolled in the high dose group, with each volunteer receiving between 7 and 9 IM injections. This group has been followed for up to 1 year. Product administration has been well tolerated. Local reactogenicity was observed in only the high dose group, with all 3 individuals in this group reporting mild pain and tenderness at the injection site. Only 1 individual in the intermediate dose group reported mild myalgias. No other systemic reactogenicity has been observed. All reported reactogenicities resolved within 7 days of IM injection. No adverse events have been attributed to AAV8 VRC07. In the low and intermediate dose groups, AAV8 VRC07 DNA was rapidly cleared from the blood, with 3 of 5 volunteers having concentrations of less than 200 copies of AAV8 VRC07 DNA at 2 weeks. In the high dose group, 2 of 3 individuals had AAV8 VRC07 DNA concentrations of less than 200 copies per mil at 8 weeks after injection. 1 individual in this group did not achieve a concentration of less than 200 copies of AAV8 VRC07 until week 28. As expected, all volunteers assayed who were given AAV8 quickly developed anti-capsid antibodies. In all individuals, anti-capsid antibodies persisted for as long as measured. In low-dose volunteers, administration of AAV8 VRC07 resulted in measurable VRC07 within the first 4 weeks after IM injection. VRC07 concentrations then decreased from 6 weeks until about 16 weeks, at which time VRC07 concentrations began to increase and then achieved a stable concentration for the remainder of the first year. Similarly, in the intermediate dose group, an initial peak in the antibody concentration was seen at 6 weeks, which then decreased until 14 weeks. A secondary peak was observed in one of the 2 volunteers. In the high dose, between 2 and 6 weeks, followed by a decrease in antibody concentrations between 6 and 14 weeks after IM injection. A secondary increase in VRC07 concentrations was observed in 2 of 3 individuals. In these 2 individuals, concentrations of VRC07 greater than 1 microgram per ml were achieved. Our preliminary data suggest that transgene VRC07 produced in vivo neutralizes HIV as effectively as VRC07 produced from transiently transfected X cells in vitro. In each dose group, 1 individual showed a blended increase in VRC07 in the primary peak, followed by either the absence of or diminished secondary increase in VRC07 concentration. In the low-dose individual, the intermediate dose individual, and the high-dose individual, a measurable anti-drug antibody response was observed. audA responses began between 4 and 12 weeks after IM injection of AAV8 VRC07 and persisted as long as measured. No audA response was measured in the other 5 individuals studied. As shown here, audA responses do not explain all of the decreases observed in VRC07. As you can see, in this 1 individual in the high dose group, there is a precipitous drop in VRC07 concentration between weeks 22 and week 40. There was no anti-drug antibody response observed in this individual, and at present we have no explanation for this decrease. In summary, this study establishes that AAV8 VRC07 is safe and well tolerated in HIV-infected individuals. Measurable amounts of VRC07 were found in all individuals given AAV8 VRC07. To our knowledge, this is the first study to show the induction of an HIV-specific broadly neutralizing antibody in HIV-infected individuals. Typically, a biphasic VRC07 serum concentration was observed after AAV8 VRC07 injection, with an initial peak in antibody production seen at 4 to 6 weeks after injection, followed by a decrease in antibody concentration, and then a secondary increase in VRC07 concentration after 14 weeks. Anti-drug antibody responses blunted the measured VRC07 serum concentrations in 3 of 8 volunteers, and finally, this study establishes that AAV vectors can be used to induce the long-term production of specific human antibodies. None of these studies get done on their own, and I have many people to thank. I particularly wish to acknowledge the work of Drs. David Baltimore and Alex Blas, who were responsible for the design of this vector. And I would also like to acknowledge the work of Adrian McDermott and Sandeep Narpala, who developed the assays which made this work so exciting. I would also like to thank the clinical staff of the Vaccine Evaluation Clinic for making this trial a reality. Finally, I would like to thank all trial volunteers for their courage, time, and effort.
Thanks very much, Joseph. That wonderful study. This is the contact information for reaching out to initiate a dialogue. And this is the conclusion of our session this morning. I will add anecdotally that during the session, a lot of times Apple Watches tell you to stand up. I'm pretty sure they're going to be programmed for us in the future to tell us to wash our hands. Thanks very much for joining us today, and thanks, Judy, for co-chairing with me as we wash our hands at the end of the session. Thanks a lot.
Bye.
Bye.